Well, it's a last session, last effort for me, but then it's bad for you. Exam tomorrow. Sorry, it's fun, but then it's coming work, it's work, no choice. So we're reaching another point. Remember, we were discussing already this point. The probiotics need to be alive. But then, if the metabolites can perform better, what's better than this? Because if you need to keep the probiotic alive, you need to be keeping the appropriate number. So all these things is actually, it's a trouble for industry, for dairy, for pharmaceutical. They need to keep this microorganism in the right number, in the right conditions. So it's kind of problem. So they actually, they look for the short way to resolve this problem. And they were saying, okay, we can go for the spore-forming probiotics. Because spore-forming probiotics, you form the spores, you keep the spores and you sell the spores, and then these spores can grow and they can perform their work. Well, but again, not all the applications can be done by the spores. And we already know that some metabolites are produced by non-spore probiotics. So lactic acid bacteria, they're not forming spores and they have their benefits. We're having as well Akkermansia is not forming form and needs to be alive. We're having Saccharomyces cerevisiae, they don't form spores and need to be alive. So this is actually a point that spore forming was not really solving the problem. So some old people had actually did and observed something interesting point, that even if you use the dead cells, in fact they can do the same job like a probiotics, because very often probiotics could be associated with the effect of some metabolites. Well, what is the definition of postbiotics? This is the preparations of inanimate microorganisms and all their components that confer a health benefits. Everyone agreed that the postbiotics are dead cells. Well, some say that can be entire dead cells, sometimes that some metabolites, but normally the general idea is that postbiotics are entire cells, everything together. Because if you have the metabolites, you have the metabolites. You have effect of enzyme, you have the effect of bacteriocin, you have the effect of polysaccharides. If you can call them metabolites. And if you apply the entire cells, you're applying actually as postbiotics. Some of the important points that as well need to pay attention, probiotic needs to be safe microorganisms, because this probiotic as a live organism will enter the cells and will be interfering with your host immune system. So it's very important that the probiotics need to be safe strains. And they need to have appropriate studies on their safety. When it's questioned about the postbiotic, in fact, the safety of the microorganism is not such a big deal. Well, I do prefer the postbiotic to be derivative of the safe microorganism, because you don't know, sometimes maybe, even if as a dead cells, you can maybe have some metabolites that could be with some negative effects. So in this point of view, the postbiotic safety is a little bit easier to deal with. And as well, we can think about the fact that we don't need to deal with the keeping this organism, because postbiotic, you need to keep it alive, having short shelf life. Postbiotic is already dead cells, so you can keep it for a longer period. So you need to be aware that the metabolite is not going to be destroyed. But already kind of conservation process is much more easy. Another point, if you think about the postbiotic, postbiotics you can easily quantify, because you quantify specific activity. You quantify enzymatic activity, you quantify antimicrobial activity, you quantify something that analytical chemistry already have enough methods to be quantified. And you have the probiotics. I'm sure that probiotic will produce these metabolites when it's inside of your body. Maybe not. You're having in the plate a very good production of short-chain fatty acids, but when this probiotic go inside your body, maybe it will not produce the short-chain fatty acids. So it's important to be sure that you're going to have this production. In postbiotic, you already have produced everything. You quantify it, you know the quantity, you know the quality, and you can apply it. And it's making the entire process much more convenient and easy for application for manipulation. So more or less, this is a paper that is, okay, I will say almost published because we had already the first revision. There was, editors was, reviewers were suggesting minor revision. So we're just waiting now the authors to confirm that paper, it's, everything's okay, and we can be accepted. So it's one step before accepted. So this I could submit it. So we have the microorganism. You need to select appropriate microorganism. This could be yeast, this could be gram-positive, gram-negative. If they have their benefits, you need to culture them. This can be liquid culture, could be solid-state fermentation, because you want a biomass, you want the cells. When it's question to culture them in the solid-state fermentation, you can use several waste products from the food industry or from the other processes. And you can actually use this solid-state fermentation applying on the very cheap substrates, and in this way you can have a cheaper way to production. When you go into a probiotic, you're aware a lot of possible contamination from the media, but in this case you can even combine all the media residue, all the cells, all the biomass, because if you are using the food residues for their growing, it's even, you can be much more easy for you. Then you need to find the way, you need to kill this microorganism. So remember the story, how to kill Listeria monocytogenes. Artificial intelligence say you're not supposed to kill. Well, we need to kill the microorganisms because we're going to apply them as a dead application with beneficial properties. So this process could be very different. Very often people immediately think about heat. Well, autoclaving, it's a kind of high temperature. We kill the microorganisms. But then we're having a big problem, because if you kill them by heat, what? Okay, if you kill them by heat, what's happening? Maybe you can destroy some vitamins, maybe you'll destroy some enzymes. So you're losing probiotic, but no, you're losing beneficial properties. So very often, this way, this step here, how to kill the live microorganism and to prepare the postbiotic, it's a very crucial, very important step. Because you need to be sure that you're not destroying beneficial metabolites. So obviously, high temperature is the easiest way, but maybe it's not going to work very well. So we can use the high pressure, because high pressure will maybe destroy the cells, but at the same time, most probably will keep the metabolites in the good condition. You can use sonification, but well, sonification can actually destroy some of the proteins. And if you're having some enzymes, they're highly molecular weight proteins, maybe sonification will have some problems. Even in some situations, sonification can maybe even have negative effect on the DNA. And if you need a DNA or RNA to serve as an inductor for some beneficial process, maybe we'll have a problem. Mechanical destructions. We can always use the glass beads and destroy the cells by glass beads, by the highly speed vortexing. We can use some chemicals, so we can use even some enzymes. Idea very often to combine all these methods in the way, then we're going to preserve our objective, our metabolites that we want to have it in the postbiotic. So then additional biotechnological approaches for postbiotic preparation must be developed, because obviously we're having the beneficial metabolites, but again, most probably we'll need to do some kind of modifications to remove some of the components and to focus on the other kind of partial purification, but still not fully purified, because if we fully purify, we work with the metabolites. And then biological and chemical assays to need to be selected products with stable biological activity and chemical properties. So now, analytical chemistry needs to confirm what kind of metabolites are they, and if they're having even quantification. So you make a postbiotic preparation, you say, okay, this postbiotic preparation have this level of the vitamins, this level of the enzymes, you need to do this part. But end of the day, you're taking advantage of the probiotic cells that but that's not obligatory. That could be not even probiotic. If they're producing metabolites, great. If you have the metabolites in the end, this is the point. And you can go far away of the problems that you need to keep the live microorganism. As well, the point, very important point, is the fact that for immunocompromised people, even the best probiotic could be dangerous. In this way, we're going away because we don't have nice cells. We are applying metabolites produced by these microorganisms. So, in fact, from the same paper, probiotics versus postbiotics, possible systematic infections. Even the best probiotic could be bad for some group of people. Colonizing, well, we want to put these cells in our host, but very often long-term colonization. What about later we discover that this colonization is not good? What we'll do with this strain is already there. And postbiotic, we don't have this problem. We have safe and more control on the application. So you're giving now, doing the job, you're giving again, doing the job. You don't need it anymore, you stop giving. Stability of these products is much more stable. They can be kept very easily and more. We know what exactly you have. In the probiotic, sometimes they can express something else. They can make attachment. When they're in the gastrointestinal part, maybe they will produce something different. Then we never think about this because specificity of the metabolic pathways can reflect and produce something different. Technologically advantages, formulations, delivery, applications, all this is very benefits. And this cartoon is like the probiotic, say to the postbiotic, die. But probiotic answer, I have never been a probiotic, but even then I will be better than you. So they can work and they can perform better. Don't say this to my chicken, because I hope she will not be watching this video. But this actually looking a little bit like him. He likes to wear this kind of spectacles, circle one. It looked like, but he drew it. So maybe he was actually playing and putting me killing him as a probiotic killing postbiotic. Okay, then some of the metabolites produced by the lactic acid bacteria that can be actually considered as a way of the, now coming to this postbiotic story, GABA. Gamma-aminobutyric acid, called as a GABA, can be produced by several lactic acid bacteria and in fact can have the beneficial properties. Well, some probiotics are already known that can express GABA even in the gut environment and then can be producing. But again, what about if we can have the control fermentation process? You have the strain, we're producing GABA in the biofermenter, and then kindly concentrate and slightly go rid of what we don't want, and we have kind of dead cells together with the GABA. So it's not a bad idea. GABA is an inhibitory neurotransmitter. It lessens the nerve cells' ability to receive, create and send chemical messaging to other nerve cells. So now a transmission of the neural signal is very dependent on presence of GABA. And many medical conditions are in fact associated with changing levels of GABA, and medications target the GABA receptor. So more evidence are needed to know how the GABA can actually modify. Well, in this moment we believe if we add a little bit more GABA in our food products, and if you're having bacteria that can produce GABA, it's good for us. But again, with development of the research and small studies, we will know better about this. By the way, in Korea, they're selling chocolates with GABA, enriched with GABA. Well, I tried, I don't feel much more better, but they say it's good for me. So, by the way, they're not more expensive, the same price without GABA, but okay, we can have it. What's wrong to have it? Okay, GABA is non-protein amino acids, so it's very important, this point. Go. Okay, normally it's associated as neurotransmitter, so the presence of GABA will facilitate transmission of the nerve signals, but at the same time for the bacteria, because GABA is important for us, but what's making good for the bacteria? Why bacteria producing GABA? It's actually helping them to resist to acidic conditions, because what's happened, come on, okay? When you have a high acidic level, bacteria need to deal with these ions. And what is happening? You're having glutamate in the presence of low pH, so presence of hydrogen ions. Glutamate is transformed to GABA. And in this way, in fact, you block the presence of H plus ions. What's the name?
Hydrogen ion.
Okay, because in Bulgarian it's different, in Portuguese it's different, in English it's different, so need to remember all these names. And normally this it's associated, production of GABA, in fact it's associated very much with one enzyme, GAT. So if you can detect this enzyme, it means the strain having chance to produce GABA. But now, we was doing this project as well to select some beneficial microorganisms that can produce GABA. So one of my students, he just defended his master and now starting doing his PhD. He was isolating strains that can maybe produce GABA from Brazilian cheeses and from feta cheese from Bulgaria. So I brought this cheese in Brazil. It's allowed. It's not against the law. You can carry the cheese to Brazil. Dairy product is allowed. Meat product not allowed. And it was scary for me to eat this cheese because I like it. But then when we start the project, okay, let's have the samples and try to isolate something. So first of all, when I was isolating the strain, we was checking through gram and catalase because we wanted lactic acid bacteria. I think actually I can sit here. I think for the camera it's okay, you can see me. Okay, a little bit tired, I mean 5 days, it's a marathon. Okay, so we was doing gram and catalase because we wanted to be sure that we're selecting lactic acid bacteria. It's easy. I mean catalase and gram, okay, gram take a time, but take to observe the strains, but I mean students needs to know how to do the gram. So we're selecting always lactic acid bacteria based, okay, presumably lactic acid bacteria. And then we wanted to know if this bacteria can maybe produce GABA. The method, someone already worked with the GABA production here in the room? I'm suggesting you to do this work, it's not difficult in generating quickly results. So in a very lazy way, in not too much work, you can have a lot of results and impress your supervisor with going on the meeting every week and showing a lot of results easy to generate. So you have the samples. Half of the samples we centrifuge because we need to obtain the cells and supernate them to separate. And then we use the pellet. On the other side, we take the samples, we sonificate them because we want to destroy the cells, to liberate if something is inside the cells. So 2 options. Then 2 samples, sonificated, sonificated sample and the normal pellet. And we use the spectrophotometer method to do this test. We use ELISA plate, 96 post plates. And then very simple method when you add buffer bronchresol pyridoxal phosphate and then you add glutamate. Because with this way you're going to have glutamate as a substrate for production of GABA. So if you have presence of the enzyme, GAT, it's called GAT, you're going to transform glutamate and you have the indicators, they're going to have a change in the color. So you put in the microplate in the microplate reader and you start reading every minute, every 2 minutes, because if you have this kind of line that's going up, it means you have production of GABA. It means the enzyme is there, it's transforming glutamatic acid to GABA. If the line is like flat, no. Well, in this project, we had quite a big number of strains, and we was trying to pre-select some of them. Again, more or less about 400 isolates from cheeses, but on one plate you can add 96, okay, you have controls, let's say 90. So make 4 plates only, and you have the results in, let's say, in less than 2 hours. So, okay, he never done in 1 day all the work, but I mean in less than 1 week he was able to see which of his isolates, in fact, have the potential for GABA production. But since we're always looking for bacteriocin production, he was as well checking what of these strains are bacteriocin producers. So after that, he crossed the data of bacteriocin and GABA production, and he selected 8 strains for the future study. Well, he selected a little bit more, but then do the DNA extraction, do the REC-PCR, do the identification, and realize that some of them are replica of same strain isolated in multiple occasions, but in the end, he has 8 strains that go further. Then question is how the GABA production is dependent of the condition. So we tried one of the time methods. So you can play with different cellular density, but you keep all the other 4 parameters blocked. So we're having as a standard 10 to 6 cellular density, pH 6, incubation time 24 hours, monosodium glutamate at 60 millimolar, and temperature 37. So if you change the cell density, all the other you're keeping the same. Then you keep just play with the pH, but you keep cell density in the other constant. Incubation 10, you do it 24, 48, 72, 96, 120 hours. But the rest of the parameters, you keep stable. You don't correct. Then monosodium glutamate is mean the precursor for production of GABA, and temperature. So what we observed, if you do this variation, you can see that the GABA production is very dependent on several conditions. So let's say you play with the sodium. Okay, this is the different strain when we selected, and this is the different conditions. So you see, in some conditions, sodium glutamate is very important. In some conditions, the time of incubation for other strains is as well one of the important moments, or temperature could be playing as well reaction. For some of the parameters, you can see it's a different condition. So obviously you don't have the same conditions for all the strains. Again, it's a bad news, you need to do it. And the problem of this process is associated with a little bit more work, because all this process needs to be done on the mass spectrometry and partial purification and quantification by the MSC mass spectrometry method. So in fact, if you do kind of generalization for this 8 strain, you can see that more or less 10 to 6, it's more important cell concentration that's important for GABA. That actually is good news, because it's a little bit below requirement for the probiotics. So if you apply these strains as a probiotic, they have a good chance to produce GABA in our gut. As well, when you look for the pH 6 neutral, it's a good news, because imagine if it was 4, it would be a problem. Because, I mean, for our gut system, it's, I mean, lower gut, it's not 4, there is like neutral, so it's a good news. And as well, glutamate concentration needs to be quite high for this optimal production. But well, glutamate can be delivered by several food products, because it's a part of the food, so no problem to deliver this. We don't need to give glutamate, pure glutamate. We're going to give food products rich of glutamate, but this way we can actually have this production. And then time of the incubation could be for 24 or very long. Well, most of the time food stay in our lower gut about 24 hours, so time enough for this microorganism to produce GABA in silico. Well, temperature, 50 degrees Celsius is the best one. So it means if you're going to biotechnological production, it will be possible to do it a little bit higher temperature, because you put in bacteria in stress, very high, not going to kill the bacteria, but it's a stress, so obviously pushing them to produce more GABA, because most probably the pH is trying to affect the strains. But again, the 37 was not bad at all. We're having the second peak on 37. So all this showing that the GABA production could be done in silico. And then with GABA, now we wanted to know in the next project what going to Kaiki Ordonio. She just started his PhD like a few weeks ago. He was actually wanted to see how the GABA could be applied, and he wanted to use the mouse model to see, because the GABA can actually have an anti-cancer effect as a component. And since some of the bacteriocins that he worked is showing a very good anti-cancer effect, bacteriocin himself. So now he wanted to use GABA and the bacteriocin in combination and use it on the cell line, and after that on the animal model, to see if he can manage to control some of the gastrointestinal cancer that could be affecting, could actually be controlled by this combination, bacteriocin and GABA. And he's going to explore this on the molecular and physiological and animal model, because idea is to having this probiotic later for the prevention, because these days we can detect possibility of gastrointestinal cancer by some markers. You do the blood test and you say, okay, maybe you're going to develop this kind of cancer, because marker is supposed to move. And for these people, they still don't have cancer. They're clean, but they're at the risk factor. So what's the best option if you can apply the probiotic that can have reduction of this cancer, and you can actually prevent them on developing of the disease, or if at least you can push it, like, I mean, in this case, even if you push 10 years later, it's even better. Because if you can prevent, it's excellent. If you can postpone the disease for later, it's another option. But again, it's prevention could be a good option. Another project that is run, we were testing different fermented food products that are normally consumed in Brazil. You see the variety of them. Some of the names may be familiar, some was completely strange names. But well, there's a variety of the fermented food products. And we were screening all of them for the potential bacteriocin producer, and as well potential producer of bioactive metabolites, with the idea to apply them as a probiotics. Interestingly, we have several very strange, lot of bacillus beside these isolates, with a good antimicrobial productions. Some of them were already tested, and we showed that they have probiotic potential. Even on the cell line, there was showing increase of the anti-inflammatory cytokines, so it's showing a good future and potential. Well, this work is going by Emilia. She is a postdoc in the laboratory. But the problem in Brazil, the postdoc, they hunting the job all the time. Because the postdoc is for 1 or 2 years and you want a permanent job. So she hunting the jobs and doing the work in the lab. So in the moment when she finds a contract and moves to a permanent job, she will leave the lab. But again, there will be another person, maybe some of the PhD will continue with this work, and we're going to complete and see which one of these fermented food products, in fact, could be more promising later to be developed as a functional food product, because we're going to enrich them with their own bacteria. And since we know what is the beneficial properties of this bacteria, we're going to know how we can apply them and what we can use them for. Well, it's everything just the angels. Now it's coming the problems with the probiotics. Well, according to the probiotic, first about the probiotic and fermented food products. Very often people say fermented food products are probiotic, fermented food products are probiotic. No, they are not. They are beneficial, functional, fermented food products. They can carry several microorganisms with beneficial properties, but they cannot be considered as a probiotic because, first of all, we don't know what kind of bacteria we have there. We know the product is good, you're feeling better, you're having stomach problem, you take these fermented food products, you feel better, but why? No one's knows. The problem is a probiotic needs to be fully identified in medical clinical study proving these beneficial properties. In fermented food products, in Daki, you have nice bacteria, but you know what they do exactly? Do you identify them? No, they are there, they are good. We know in general they are good, but they cannot be called probiotics. Like kombucha. Who like kombucha to drink? Well, it's strange fermented tea. Chinese like it. They advertising a lot, but it's not bad, but still. There's several beneficial microorganisms. Some of them are lactic acid bacteria, some of them are acetic acid bacteria. They have several yeasts. But again, we don't know the identity. And we know their number, exact number. To be a probiotic needs to be reaching us with specific number. So taxonomy not identified, not scientifically proven specific beneficial properties, or neither confirmed by animal or human trial studies. And the presence of appropriate levels of viable cell, all these things actually clearly stating that fermented food products cannot be called probiotic. This is why several dairy industry, they stop actually advertising their fermented food product, yogurts, etc. as a probiotic, because this is a specificity of the point. Even if you look here, if you make this kind of study and you put the kombucha and see what people associate with the kombucha, they say it's a probiotic very often, but you see functional food, probiotics, microbiota, health, bioactive compounds, antioxidant activity, green tea, organic acid, polyphenols, phenolic compounds, all these things. But this word, probiotic, cannot be linked to the kombucha because kombucha is not a probiotic. You can make same kind of analysis online, and you can generate the figures for any functional food products, any fermented food products. You will be surprised that several papers still associated fermented food products and claiming that they are probiotic. But it's not true, it's wrong, because they don't fit the definitions. What about the other side of the story? Several cases, probiotic products on the market. You see, till now was like a wrong perception. The industry even sometimes playing and they climbing the probiotic, something that is not probiotic. But now you have the pharmaceutical products that you buy even in the pharmacy, and they say it's a probiotic. Okay, thank you very much. They have the clinical studies that prove this strain, specific strain. In fact, it's a good. The problem very often, these pharmaceutical products, they contain a wrong strain. Someone was working with commercial products and mentioned that very often they isolated different strains, different species. Not strain, even species. Sometime on the label they say it's a single, well, most of the time it's single culture. But you start isolating, you realize there's 3 or 4 or 5. You don't not even to do the sequencing, because when you observe the colony, when you do isolation, you realize they have 5 different type of colonies. So obviously it's representing 5 different strains. This is one of the biggest problems. Very often, even the good quality company sometimes can happen to have the wrong, not more than one or even the wrong strain. There was a case, I'm not supposed to say the name of the company, but neither will say the country when this was happened, but the probiotic on the market, in fact, was contaminated with salmonella. You want to have it salmonella? Yeah. So you think that you're taking something that is good for your health, and then you're having medical, more serious problems. Containing low number of the probiotic lactic acid bacteria. Not only lactic acid bacteria, it can be yeast, it can be new generation. Very often because of the wrong packaging, very often about the wrong storage, very often because of the wrong production, commercial probiotics can have the low number. And again, it's not fair. I'm paying a quite expensive price for these probiotics, and I will take it, and then I'm having nothing. One of the studies that I was performing, every probiotic was cost about $40 every package. Imagine you're buying this and you're taking, you realize it's not appropriate number. Containing dangerous bacteria, including potential human pathogens. This story with the salmonella inside the capsules. Well, the company actually acknowledged they had a problem in their production line, and they asked us to make this check what they have it. Well, in a couple of days, he said you have salmonella. So there was stopping entire production, cleaning the line, doing all this. But I don't know if they remove everything from the pharmacies. I don't know. They're supposed to be, but who knows? Sometimes probiotics containing nothing. It's happened. We was checking some capsules, nothing. Like a sterility test, nothing available inside. One probiotic that is good for me maybe is dangerous for you, or one good for you is dangerous for me. Sometimes missing this information, they don't say who can take, who cannot take. Sometimes it's even not testing these things. And commercializing something like a probiotic that is not at all probiotic. There was even some opportunistic companies, they putting on the market even nothing. They just put empty capsules and say it's a probiotic because they can sell it. Well, regulation in several countries is getting stronger and stronger. Let's say in Brazil, in order to commercialize a probiotic, you need authorization. You need to provide portfolio. They're asking they want to ask all the studies, animal studies, in order to allow you to this as a probiotic. They're not going to. But again, remember about the contaminated with salmonella. They cannot sell products in their country. They just there is no regulations, and this is not fair because I mean playing with the health of the people. So and produce a right and deliver this product to the people. Again, probiotics same species that even on the label the same strain products, we obtain that it's okay. You see the strains are not the same. They are showing they're different and cannot be the same species. So it's not the same strain. There's some papers on this showing, and this is like 20 years, but it's clearly several probiotic products don't match what is on the label, what is inside. It's getting continued 20 years later. We have several examples that the products on the market was not really corresponding what do you have inside. Then you're having this dairy product. It's okay, the company is already not on the market, so we can talk about them. They changed their name. There was providing this kind of dairy. It's like a yogurt with fruit juice. It's a very nice and tasty. It's refreshing. They was having with different options of fruit juices. But again, if you look, I hope I can manage to do this. They have the simulating functions of the immune system. They're climbing this on the label. They're playing well with the lactose digestion. For the people that with lactose intolerance, and well, the product you can easily see from the website is from South Africa. Africans, they are quite problem lactose intolerance. Inhibit the growth of harmful bacteria. So what do you understand by harmful bacteria? It's not really clear. And then clearly stated contain probiotic culture. Well, everything sounds very good to be true. So I was enjoying this drink, and I decided let's check what kind of probiotic they have inside, because my idea was to use this culture as a control culture. Well, bad news. No lactic acid bacteria presence in this product. But we have Staphylococcus aureus growing from this product. So you see, they claim that they have the probiotic. They claim several good things that can happen with this. It is taken from their website. But again, I think it's not anymore existing, this product on the market. Neither the company is existing anymore. So there was claiming several beneficial properties, but at the same time, there was not having nothing. It's like playing with the sense, because the people see this probiotic culture and they will buy it. And very often it's more expensive than normal other products. So it's again something that I'm criticizing the system that, okay, now let's keep it this quicker. Sorry, it was Indian company. They get the problem with their probiotic properties. They were selling this in South Africa. But after that, they discovered that there was some problems. They were taking really good action to remove the products from the market. So, but this is like 20 years ago. These days, the company needs to play more careful because they actually need to be sure, because the customer is much more aware of their right. They know much more better what is product. They know much more better how you need to control, how you need to play with it. Because if not, you can having a really a problem. Customers calling and asking, you provide me something that in fact is not working very well. Many commercially available products are incorrectly labeled. We're supposed to label the probiotics in the right way. Regulation says not only to put the name of the bacteria that is inside, but you need to put even the strain. Because again, sometimes the regulation agency, they authorize a specific strain. They don't authorize the species, they authorize specific strain. Frequently